feeding state profoundly influences how animals make decisions, shaping even behaviors unrelated to feeding itself. But here's the twist. How exactly does hunger or satiety tune the brain's circuits to prioritize certain actions over others? This question lies at the heart of a fascinating study exploring how feeding states bias sensorimotor choices in Drosophila larvae, the fruit fly's young stage. Throughout the animal kingdom, internal physiological states like hunger and thirst act as powerful drivers of behavior, because maintaining homeostasis is vital for survival. Insects, mammals, and many other creatures change their responsiveness to sensory cues when food deprived, suggesting the brain assigns different values to stimuli based on internal needs. However, the precise neural mechanisms that allow such flexible behavioral decisions, especially in non-feeding related contexts, have remained largely mysterious. Survival demands that animals balance drives such as finding food with urgent needs like avoiding danger. Avoidance behaviors are instinctive and robust, but must be adaptable. A hungry animal might tolerate greater risk to access nourishment, while a well-fed animal might prioritize safety. This dynamic balancing act makes avoidance an excellent window into how the nervous system integrates internal states with external demands. At the neural circuit level, this behavioral flexibility is thought to be orchestrated by neuromodulation, substances like neuropeptides that fine-tune synaptic connections and neural activity to bias one outcome over another. Another possibility is that internal states reroute information through alternate neural pathways, adjusting processing according to context. Importantly, certain circuit motifs, such as reciprocal inhibition between inhibitory neurons, have been proposed as ideal architectures for enabling this state-dependent flexibility. But until now, direct experimental evidence demonstrating these mechanisms and neuromodulators in action has been scarce. This study focuses on Drosophila larvae, a model organism uniquely suited for detailed circuit mapping and neuron-level manipulation. Their nervous system's connectivity can be mapped at synaptic resolution, allowing scientists to link specific neurons and circuit motifs directly to behavior. Further, many neuropeptidergic pathways in Drosophila are evolutionarily conserved, enhancing the relevance of these findings across species. The researchers concentrated on the larval response to a mild mechanical stimulus, an air puff, that typically triggers competing behavioral actions: hunching, a protective, startle-like response, and head casting, an active exploratory behavior potentially linked to escape. Prior studies identified neural circuit motifs, including reciprocal inhibition of inhibitory neurons that mediate the competition between these behaviors. By leveraging advanced calcium imaging, neuron-specific manipulation, automated behavioral tracking, and computational modeling, the authors examined how short-term changes in feeding state reshape this behavioral competition. They established protocols of 90 minutes of food deprivation or sucrose-only feeding in larvae to induce measurable behavioral changes while avoiding long-term physiological alterations. Both starved larvae and larvae fed with only sugar moved faster, crawled more extensively, and explored environments more broadly, consistent with an increased drive to find missing nutrients. Importantly, refeeding reversed these locomotor changes, demonstrating the effects reflect motivational states rather than permanent changes. Interestingly, starved larvae increased intake of yeast-rich food, protein source, and standard food, nutrient-rich, reflecting nutrient deficits. In contrast, sucrose-only fed larvae increased yeast intake, but not standard food, and drank significantly more water, likely as a physiological response to osmotic stress due to high sugar intake. In a sucrose preference test, sucrose-fed larvae showed reduced sucrose preference, implying a shift in reward valuation, possibly mediated by internal energy sensing rather than taste alone. Correspondingly, hemolymph glucose levels rose significantly in sucrose-fed larvae, supporting this internal feedback mechanism. When exposed to the air puff, larvae's sensorimotor choices were noticeably affected by feeding state. Sucrose-fed and starved larvae displayed fewer protective hunch and static bends, and more active head casts, indicating a bias towards exploration or escape rather than defense. These behavioral shifts persisted even when the duration of food deprivation or sucrose feeding was extended. Importantly, calcium imaging showed that chordotonal sensory neurons, which detect the mechanical stimulus, responded similarly regardless of feeding state. Optogenetic activation of these sensory neurons produced comparable feeding state-dependent behavioral biases. This suggests that feeding state affects sensory motor decisions not by modulating sensory input strength, but by acting downstream within the neural circuit. Focusing on the neural circuit processing these inputs, calcium imaging of basin 1 and basin 2 projection neurons, previously linked to hunch and headcast actions respectively, revealed that feeding state differentially tunes their activity. Basin 2 activity, associated with headcasts, was significantly increased in sucrose-fed larvae at most stimulus intensities, while basin 1 activity was only mildly affected. Starvation showed subtler effects. The core of the decision circuit involves two inhibitory interneurons reciprocally inhibiting each other, known as griddle-2, promotes hunching, and handle-B, inhibits hunching and promotes headcasting. The study found that these neurons are modulated differentially by feeding state. Sucrose feeding and starvation reduce griddle-2 responses, but enhance handle-B responses to mechanical stimuli. This reciprocal inhibition motif is modulated so that the safety-promoting neuron, griddle-2, decreases activity, while the exploratory promoting neuron, handle-B, increases activity, yielding a bias toward exploration. Computational modeling of this circuit confirmed that reducing the maximum activity of griddle-2, or increasing input to handle-B, expanded the parameter space favoring headcasting behavior, accurately reflecting empirical observations. Further experiments showed that these inhibitory interneurons functionally inhibit each other, consistent with reciprocal inhibition. Silencing handle-B enhanced griddle-2 activity and vice versa, although the effects varied depending on feeding state. Such nuanced modulation suggests internal states influence circuit dynamics, tuning the resulting behavioral outputs. What drives this feeding state modulation? Connectome analysis revealed that the feedback inhibitory neuron, handle-B, receives input from a pair of long-range descending neurons expressing neuropeptide F, NPF, a homologue of mammalian neuropeptide Y, known as a hunger signal. These NPF neurons become more active under both starvation and sucrose-only feeding conditions. Silencing NPF neurons reduced handle-B responses and impaired feeding state-dependent increases in exploratory locomotion, implicating NPF signaling as a crucial modulator linking internal state to circuit activity. Handle-B neurons express NPFR1, the receptor for NPF, and downregulating NPFR1 in these neurons abolished their state-dependent modulation, underscoring the specificity of this pathway. However, griddle-2 neurons do not receive direct synaptic input from NPF neurons, nor express NPFR1, indicating other neuropeptides mediate feeding state effects on them. This led to exploration of sNPF, short neuropeptide F, another analogue of mammalian NPY with widespread expression in the larval nervous system. Immunostaining revealed both handle-B and griddle-2 neurons express the sNPF receptor, sNPFR1. Functional experiments demonstrated that knocking down sNPFR1 in handle-B increased its response to mechanical stimulus, while sNPFR,